If you look back through the history of science, things change in big leaps. Somebody has a really novel way of looking at something, and then suddenly it's, why hadn't we seen that before? It's obvious, it was there all the time. I mean, to use a very glib example, in the apple falling on Newton's head, I mean, why hadn't anyone noticed gravity before that?
It may appear that the human body has nothing left to hide from modern medicine. With a countless variety of mechanical scanners and chemical tests, science has stripped our bodies down to the bare molecular essentials. All this knowledge can create a false impression. In fact, mysteries and unanswered questions run deep through the history of medicine. But recently it was discovered we'd missed something very basic, a molecule that allows us to understand some of the most essential functions of the body. The first clues came from basic research that in some places was so basic, it was positively obscure. Every week for years, Professor John Gillespie visited his local abattoir in Glasgow to collect tissue samples. He was particularly interested in a muscle in the bull's penis. Not surprisingly, whenever he tried to explain his research aims to non-scientists, he always ran up against the same problem.
When we first moved to work on the retractor penis muscle, I had a grant from the MRC at that time to do this work. And of course, the MRC announce all their grants, and the media look to see what looks interesting to them. And this must have caught their eye, because I had someone from the tabloid press phoning me up, and he was quite truculent, really. And his opening was what is this I hear that you've got £75,000 to investigate the love life of the bull? And I must say that took me aback at the time. Thankfully, I'm sure I simply bored him into disinterest in the whole story, which never appeared in the newspaper, as I very earnestly explained to him the problems of this and Nank nerves.
The Nank nerves that so bored the journalist are not just a feature of the bull's penis. All mammals, including humans, have a network of these nerves, highlighted here in yellow, snaking through their bodies and supplying many of the internal organs. Bodily functions, such as relaxation of the lung or the expansion of the stomach to allow food in, rely on a chemical signal or neurotransmitter released by these nerves. There were plenty of guesses about the identity of this chemical, mostly the sort of large organic molecules the body usually used for signaling. But the truth was that although the Nank system was discovered over 100 years ago, no chemical had ever been found. Despite its fundamental role in the human body, medicine had to accept the Nank signal was a complete mystery. This was just the sort of problem a researcher like Gillespie enjoyed. He studied the penis muscle because it was particularly rich in Nank nerves, and the bull's because it gave him plenty of tissue to work with.
We had to establish its pharmacology. So this is looking at the muscle in an organ bath in which you keep it alive in saline and provide it with oxygen and nutrients. And you can then stimulate the nerves electrically and see the responses.
By doing this, Gillespie managed to isolate the mystery substance, capture in the test tube a chemical that had the same effect on the penis muscle as the Nank nerves. It was clear he was onto something.
There's something about novel substances that excite pharmacologists. I was approached this as a young chap who's just completed his honours degree, and was looking for a career in research, and I found a wonderful opportunity to work with Professor Gillespie. The substance was a very unstable substance. Now, that pleased us. Initially because we knew that the neurotransmitter was inactivated very, very rapidly, and the substance that we had isolated was very, very unstable. The problem was, though, that with such an unstable substance, it was really very, very difficult to try and elucidate its chemical nature.
All they knew about this elusive compound was that it could relax muscles. While the Scottish team continued their research on nerves, another mystery chemical had surfaced elsewhere in the body, this time in the blood system. For years, drug companies and research groups had been investigating what controls blood pressure. Their aim? A new treatment for hypertension and all the enormous clinical benefits and profits it would bring. But in 1980, a discovery completely shifted the emphasis of this work. The researcher behind this was Professor Robert Furchgott. At an age when most of us would be thinking of retirement, Robert Furchgott is producing more papers than ever and doesn't believe you can ever be too old to research.
If you're lucky, you can come up with a good idea at any age. I think I was very lucky to happen to be at the right place at the right time with the right background.
Furchgott's research career was given a baptism of fire during the Second World War, when he was asked by the American government to investigate a major killer on the battlefield. A condition known as wound shock was killing soldiers even after they were rescued and treated in hospital. Unfortunately, the science of the 40s was unable to find out why, but it ignited in Furchgott a lifelong interest in the physiology of the blood system. He developed a method of studying the vessels by keeping small bits of artery alive in an organ bath and measuring on a graph their response to different chemicals. But strangely enough, it was only after he discovered what, for decades, he had been doing wrong that his mystery compound turned up.
Well, when we were making the preparation, which we had started at some time back, way back in the 50s, of a rabbit aorta, we used what we call the helical or spiral strip. We would cut the aorta into a spiral, but in doing so, we would pull the inner surface, you might say accidentally, over a finger, and that, unknown to us, was rubbing off the endothelial cells.
The endothelium is the thin inner layer or lining of the blood vessel. Damage it in any way, like rubbing with a finger, and the vessel loses its ability to relax or vasodilate. In a series of classic experiments using intact tubes of blood vessel, Furchgott showed that this delicate layer was in fact producing a chemical that relaxed the muscles around it, expanding the vessel. This so-called endothelium-derived relaxing factor, or EDRF, suggested a new local approach to the treatment of hypertension.
We had found something totally new in terms of how a vasodilator works. It was a mystery to us for many years, more than we're a little bit embarrassed to say how many years. And it was a mystery to other people too. There were some other guesses about what it might be.
While the competition to find EDRF intensified, back in Scotland, some advances had been made with the other mystery compound. John Gillespie's team had found that its potent ability to relax muscles could be blocked by the blood chemical hemoglobin. Billy Martin saw an opportunity to cross-fertilize ideas.
I wrote to Professor Furchgott. He knew that we had isolated a substance, and I took the knowledge that I had gained in Glasgow to New York, and certainly that paid off really very well. There was almost a revolution worldwide, and here was a new phenomenon discovered, and the numbers of people working in the area just mushroomed enormously. And certainly there were a lot of very, very clever people in this field, and the field was moving very, very quickly.
It's quite good to see your best students go off. We would quite like, in the end, to see some of them come back again, but we continued to discover various bits and pieces. So we were gradually building up a portfolio of information on the compound.
We had found, especially when Billy Martin was working in my laboratory, that the relaxing effect of so-called EDRF, endothelium-derived relaxing factor, could be blocked by hemoglobin added to the bathing solution around the muscle.
Billy Martin's contribution provided a useful clue and allowed Furchtgott to dismiss some candidates for EDRF. But the compound itself disappeared too quickly for researchers to pin down, and without it, they were still left guessing.
There were several groups that were trying to find out what it was, more than several. And there was some bad guesses, just as our first guess was not a good one.
The next clue to the identity of EDRF came from a very different field. Amyl nitrate, or poppers, had become popular during the 60s because of the instant buzz they provided. Poppers are, in fact, part of the larger family of nitrate drugs whose powerful effects on the human body were discovered in the last century. They've been used ever since in the treatment of angina. Despite all the work over the years, it wasn't until the 70s that drug researchers discovered nitrates worked by releasing a simple gas called nitric oxide. Once free inside the body, this gas passed into our cells, raising the levels of a key messenger molecule called cyclic GMP. Drug researchers like Lou Ignarro were then drawn into the hunt for EDRF. Immediately, he found EDRF also pushed up cyclic GMP.
This increase in cyclic GMP levels mimicked what we had found earlier with nitric oxide. Authentic nitric oxide gas also produces an increase in cyclic GMP formation in the blood vessels that it relaxes. I think you have some good ideas that perhaps... One of my graduate students in the laboratory suggested that, well, gee, maybe EDRF is nitric oxide. And I told him that he was crazy and that we'd have to embark upon a different direction.
Vial or in a containing compound rather than nitric oxide itself.
1, 0, ignition, liftoff, 30 minutes after the hour.
Ignaro's response was not surprising. Nitric oxide, or NO, was nothing like a chemical the body would use. In space, NO can be seen. It causes the mysterious aura called shuttle glow that wraps the shuttle as it orbits. On Earth, this combination of nitrogen and oxygen is a pollutant created by high-energy reactions like lightning or man-made processes like the internal combustion engine. Once formed, this tiny molecule, the 10th smallest known, is unstable. The pair of atoms prefer to grab another oxygen and form NO2. NO itself only lasts seconds. In the mid-60s, it contaminated cylinders of anesthetic gas and killed a patient before the cause was identified. The idea that our cells were actually manufacturing this killer gas was crazy. NO itself was dismissed as a candidate, and in organ baths all over the world, EDRF continued to frustrate and confound the scientists competing to capture it. More laboratories joined in, including that of Professor Salvador Moncada in Beckenham, Kent. He found a way of persuading the elusive EDRF to stick around longer by adding another compound called superoxide dismutase, or SOD.
What SOD did was to enhance the half-life, to increase the duration of the life of the substance, and therefore we could see it for longer. Of course, in EDRF terms, this is very relative because EDRF has a half-life of 3 to 5 seconds, and we probably were increasing it to 5 or 6 seconds.
Still not long enough to pin down EDRF. But when Moncada published his results, the chemical reaction he described struck a chord across the Atlantic. Professor Furchtgott became convinced his search for EDRF was over.
I was in this plane coming back from a meeting. I started to think about superoxide dismutase, stabilizing the action of EDRF. And then I began to write some equations, as far as I could remember, about the reaction of nitric oxide and superoxide. And on paper, anyway, the reaction is the two react and form nitrate, which is quite an inactive species. And I thought, well, maybe this is what's going on.
EDRF and NO resembled one another very, very closely throughout all of the experiments that we had done. And I still insisted that the cell could not make such a toxic substance as NO, and I would conclude to my laboratory people that perhaps EDRF is related in some way to nitric oxide, but it just couldn't be this small gaseous substance. And then finally, after certain critical experiments that were conducted, I decided to change my views. I mean, data never lie. All the data pointed that to the suggestion that EDRF was in fact nitric oxide.
At a meeting, both Furchtgott and Ignaro decided to go public with their radical new theories. Of course, they didn't know about each other.
Oh, I wanted to startle them. The only person I told about it before the meeting was Billy Martin, who had come and was a house guest for a day or two before we went out to Rochester, Minnesota, to this meeting on mechanisms of vasodilatation. I just got up and talked about it. There were varied reactions. I think most of the audience was completely taken aback. One person wasn't, that was Lou Ignaro, because he had his paper coming a short time after mine also proposed that the EDRF was nitric oxide.
In a way, I was a little bit disappointed because I thought that my presentation of a novel hypothesis that the EDRF was NO sort of was diluted by the fact that someone else had hypothesized it at the same time. But then, on the other hand, I was delighted because I was sort of nervous to present the hypothesis in the first place that EDRF was NO. I thought everyone would think I was crazy, but certainly they wouldn't think that two people were crazy.
It all seemed to fit together very nicely. I was immediately excited by that possibility. And I thought it was a fascinating idea.
I've known Salvador Moncada for some time, and I couldn't help but note that when I was giving my presentation, he paid a great deal of attention to what I was saying. I won't forget he was sitting at an 11 o'clock position up to my left and very busily taking notes and very quiet about the whole thing.
Moncada realized that all this was still only theory. Furchtgott and Ignaro were convinced, but the idea was still crazy. To convince others, what was really needed was for someone to actually measure living cells pumping out this lethal gas.
Suggesting something very interesting today. What I did was to phone my collaborator, Dr. Richard Palmer, with whom we had been working from the beginning. I told him what I had heard. I told him that I was very excited. We wanted to measure the nitric oxide gas by itself. And we started to look for methods to measure nitric oxide directly and found that there is a machine that has actually already been developed and is used in the car industry to measure nitric oxide levels in the exhaust of petrol engines. That machine can measure directly nitric oxide, and we thought that that's the machine we wanted. Richard Palmer traveled there and took the cells with him one day, stimulated the cells, connected to the machine. And sure enough, every time he stimulated the cell, he could see a little peak of nitric oxide generation. But it was very small. Those little blips in that machine were enough to convince us, but we had to convince other people. So we got in touch with our workshop at Wellcome and discussed the problem of making a machine more sensitive than the one in Surrey. And he set out to do this. Within a couple of months, he had produced a machine which was about 1,500 times more sensitive than the one we had used. And when we did the experiments in that machine, we could get the very nice tracings that we finally produced.
There it was, living cells pouring out a toxic gas quite naturally, controlling blood pressure, right at the center of physiology. No lightning, no high energy. Somehow, animals had evolved to harness this toxin for their own use. But was it just a rare ability confined to higher animals? Moncada decided to hunt for NO production elsewhere on the evolutionary tree.
We knew that to test the hypothesis, we needed something very primitive. And we came across this species, which is called the limulus crab, or the horseshoe crab, which hasn't changed in evolution for the last 500 or 600 million years, and has a very primitive cardiovascular system. And we found that the cells in this system have the ability to generate nitric oxide like the mammalian cells. And therefore, this must be a system that goes back in evolution at least 500 million years, and perhaps more.
It looks like NO has been with us since primitive life began, a combination of the two most common gases, lethal when made in the environment, but safely utilized inside animal bodies. It was unlikely such an ancient process would be confined just to blood vessels. Soon after Moncada published, one researcher from Liverpool decided to take a second look at a mysterious type of nerve cell that was spread throughout the brain.
The meaning of these cells at the time was totally unknown. And in fact, people just forgot about that for 20 years, paid no attention to that finding. And it only started creeping into the literature about 10 years ago, when it was found that in certain disease states, chronic types of disorder affecting the brain, you could get some bits of the brain where virtually all the nerve cells would be wiped out. They'll have died off. But those particular ones which stained in this funny way survived.
Using rat tissue, John Garthwaite managed to isolate the chemical these cells were producing and began to look at its characteristics.
Sure enough, there was something there which walloped up the levels of cyclic GMP. There we had our factor, and we showed then that it decays extremely rapidly. Within a period of seconds, it disappears. So it's a very unstable sort of messenger, totally unlike anything that had ever been described before in the central nervous system.
Unstable, increasing cyclic GMP, this mysterious neurotransmitter sounded familiar.
I rushed over to an adjacent department where I knew they were doing these sort of things with blood vessels, and I talked to some people there and saw the sort of apparatus that they had to do this, and this kind of huge organ bath type of setup, which was going to be totally inappropriate. So in order to do the experiment, what we had to do is to miniaturize everything. So you ended up with a tiny little bath with a tiny little bit of muscle from a blood vessel in, and if we add the stimulus, which normally stimulates brain cells to release nitric oxide, if we add that just to the muscle on its own, nothing happened, as we expected. And then what we did is to add in some cells from the brain into this little bit of muscle. And then, lo and behold, when we added this thing which excited the brain cells, lo and behold, there was a nice relaxation of the muscle.
These brain cells were relaxing the blood vessel because they too were pumping out NO. But what stunned Garthwaite's colleagues was the idea that NO was some kind of neurotransmitter. it meant nothing less than rethinking how the brain works. After all, NO is such a messy way of signalling.
Nitric oxide breaks all the rules because what it does, it's produced by neurons and just simply diffuses out in all directions from where it's being made. So it can influence cells which are sending signals, it can influence cells to one side or the other of those connections in a way which we hadn't expected to find in the brain. Seemingly a disorganized fashion, a random signaling across a three-dimensional volume of brain tissue, rather than this nice point-to-point signaling that we've grown to understand was how nerve cells send messages.
NO was beginning to look like a very awkward discovery indeed. But the researchers knew they still had to answer one more question. How was NO being made without vast amounts of energy? Where, among the complex chemical pathways in the cell, was the one that made NO? None of the known ones fitted the bill. Without that pathway, the story was still incomplete.
We were looking for a biochemical mechanism of how nitric oxide was actually synthesized. And we had several hypotheses of how it could be made by the cells. But going through the literature, we came across the papers of Marletta and Hibbs.
The papers Moncada found were nothing to do with blood or nerves. They came from a scientific puzzle in a completely different area of physiology. Animal metabolisms were not meant to be able to manufacture nitrates and nitrites. Yet when a measured amount of nitrogen was put into an animal, frequently more came out the other end.
There didn't seem to be any major driving force for mammals to make nitrates. You can often look at metabolic pathways in animals and say they're doing this because they need energy, they need the molecule for some life process. There was no reason for us to need nitrates. There didn't seem to be any rational reason to have them be breakdown products of anything else.
In the 1980s, in a series of studies on humans, Mike Marletta and his colleagues confirmed that despite this, we were making nitrates. But where?
Knowing that mammals make nitrates and knowing that humans make nitrates is relatively easy first step, but to decide where they come from is like looking for a needle in a haystack. But we got lucky, and there was one of the human subjects became ill. In fact, got a very bad case of diarrhea, but continued to collect his urine, and the nitrate level went way up during the time when he was sick. There was the key. That was a key that said that the immune system could be involved.
Using mouse cells, they traced the production of these nitrates to the white blood cells or macrophages. These large mobile cells are the soldiers of the immune system and possess lethal killing abilities. But what did this have to do with the production of harmless nitrates? And where did their anti-cancer abilities come from?
Part of that is chemical intuition, of knowing how biology and biochemistry works. And we came up with a guess, we like to think it was an educated guess, that it was a free amino acid that was responsible for nitrates. In other words, the nitrates were derived from a free amino acid. Arginine is one of the 20 naturally occurring amino acids. It wasn't the first one we tried, it turned out to be the third one, but by trying it, what I mean is that if you took macrophages and took away the arginine, now we have macrophages in culture, if you took away the arginine, they wouldn't make any more nitrates. If you gave them back the arginine, they did. That's how we figured out then, in this pathway now that we're trying to build up and understand, that macrophages would take arginine and convert it to nitrates. We knew that nitrates were produced, and we knew that macrophages could make them from the amino acid arginine. That's what we knew. We also knew something very important, and that was that macrophages could kill tumor cells. We didn't know how, but we knew that they could kill tumor cells, something that they were making. And it was Hibbs' work that showed that arginine was required for that killing. So it's very compelling then to tie together the new metabolic pathway with the ability to kill tumor cells. The question was, the question mark here, what was it? What was killing tumor cells? And that answer for that really came from the EDRF field, from Moncada's work that showed that endothelial cells could make nitric oxide. Nitric oxide's not stable, decomposes to nitrates. So that seemed to be the answer. And so we can erase this, replace it now with nitric oxide. That is indeed the compound that's produced to kill tumor cells.
Marletter's group had found a completely new pathway in the cell. They had also found another part of the body making NO, this time not as a signaling agent, as in the brain or blood, but a lethal defense weapon against cancer cells and invading parasites. Suddenly, the biochemists realized they had stumbled across something big.
What finding that endothelial cells made nitric oxide expanded the horizons well beyond just the killing of tumor cells. Instead, brought us into a whole new area of how cells signal one another. The EDRF signaling is a very different story. Lower levels of nitric oxide, it's not toxic, and it's being used by the cell for one cell to talk to another one. There are two sides to nitric oxide. Just as with anything, there's a little bit of something could be okay, and a lot of something could be a problem. Macrophages in the immune system makes a relatively large amount of nitric oxide in a defined area, namely in an area where there is an infection or where there's a tumor cell or tumor growing. And it makes it in relatively high concentrations. And there it's toxic. If it made that amount throughout your whole body at one time, it would certainly kill you. But it's making a high concentration, but in a defined area. When it's used as a signaling agent, precious little is produced because it's very potent at what it does and how it functions. So precious little can lead from one cell producing it, communicating with another cell that's the target.
The NO discovery showed that deep inside our cells, we have a Jekyll and Hyde molecule working throughout the body under strict biochemical control. Basic research had done its job and found a new pathway. Now to apply this discovery. But the problem was, all the work until now had been done on animals. The effects of NO had never been tested in humans. The key was a chemical inhibitor that the biochemists had already developed.
Having this inhibitor allowed us to shut off the synthesis of nitric oxide and to shut off then all the effects of nitric oxide. So having it in hand allowed pharmacologists studying blood vessel dilation to add the inhibitor and to see what the effect was.
But that was all in the test tube. What would be the effects on the first human guinea pig?
So many times in the history of medicine and pharmacology, there are experiments in animals which look very, very exciting. A lot of work's done in animals, and they move to humans at a rather late stage. And at that stage, you find out that the whole story is different, or it doesn't matter.
Patrick Vallance had only just started his career in clinical research, but he immediately spotted NO's importance. If NO relaxed blood vessels in humans, then blocking its production should lead to an increase in blood pressure. At first, he tried a tiny amount of the inhibitor in the veins of volunteers.
I had personal interest in the experiment, so I volunteered immediately for one of the infusions. I think it was very important to do the experiment to see how it correlated with the animal observations, and very exciting when we actually saw what was happening.
If you're not happy to have the infusion on yourself, then you shouldn't be happy to give it to someone else. So when we're experimenting here on basic mechanisms, we tend to look at each other first.
Just rest your head back.
The results looked promising. So next he moved on to larger amounts infused into the arteries of a whole arm.
I selected very low doses to start off with, which were too low to really do anything. And then gradually I increased the dose until we saw an effect. I remember it very well. the infusion was going in, blood flow fell by about 30%. Now that's equivalent to blood pressure increasing by about 50 or 60%. So all of a sudden, I knew that the experiments that had been taking place in animals were going to be of relevance to people. I think the discovery of nitric oxide has really completely revolutionized the way we think about high blood pressure. In the past, it was always thought of as an increased constrictor tone. In other words, there were things which were contracting down the blood vessels, and that's what put your blood pressure up. But we now know that it could equally well as be due to a loss of the dilator substances. A loss of nitric oxide would cause the blood vessel to clamp down and will put up the blood pressure.
Nitric oxide is a crucial factor in keeping the blood flowing. Not only does it expand the vessels, it also helps prevent clot formation. So encouraging was this work that the team at St George's Hospital decided to go one step further. In the intensive care wards, there was a lethal condition which theoretically their new knowledge of NO could help combat: septic or toxic shock.
From my point of view on the wards and intensive care unit, septic shock is a real killer. It develops by a fairly innocuous infection initially, perhaps, and then goes on to become a much more progressive, malevolent disease. You lose blood pressure, you don't get any blood to, say, the kidneys, the liver, and consequently their function falls off, and they stop working, develop renal failure, and cerebral perfusion or brain perfusion drops off, and ultimately you can, well, 80% or so can die from this disease.
Several early experiments in animals suggested that a vast overproduction of nitric oxide was responsible for certain models of septic shock. In other words, animals that were injected with certain things such as endotoxin to make them equivalent to human septic shock. The blood pressure fell, and that seemed to be due to a vast overproduction of nitric oxide.
In theory, the inhibitor could stop this overproduction, but it was very much an experimental drug, and Dr. Vallance knew the dangers of shutting down all NO manufacture.
We were now proposing to give larger doses into the whole body of people with septic shock. And we discussed this with the chairman of the ethical committee here as to how we should go forward. And we selected a group of patients who had a predicted death rate of 100%. So we were going to those people who were dying on the intensive care unit, trying a new approach to say, well, could this make a difference if we give this compound, if we block nitric oxide in these patients? So initially, we just selected 2 or 3 patients who, in the opinion of the intensive care staff, were very likely to die, and we gave them the treatment to start off with.
Early last year, Jean Gregg developed toxic shock syndrome after using a tampon.
I can remember falling on the bed just before the doctor came to see me, and then she said that you must go into hospital. I was having trouble breathing by then, and I can remember the ambulance men coming, and I said I can't breathe, and they put the oxygen mask on me, and getting into the ambulance, and that's the last thing I remember.
She was profoundly ill. She had very little, about low blood pressures, maybe blood pressure of about 60, 70. Yours and mine would be about 120. Not passing any urine, and really was very, very ill, at death's door almost.
I can remember dreaming that I was dying. I could hear people talking and saying that you are going to die. It was very, very weird, very strange.
Because of the immensely low blood pressure, we had her on lots of adrenaline, particularly noradrenaline, which is a very active agent, and we had her on enormous doses to try and maintain her a central blood pressure, if you like. But to do that, it has to squeeze blood from the peripheries, and so it shut down all her small vessels to her fingers and her toes. Consequently, it was a sort of inducement of gangrene. And in fact, her fingers and her toes went very, very blue. But that was at the sort of the expense of maintaining a central blood pressure. And even then, we weren't able to maintain that very well either. Things were just progressing and getting worse and out of control, in fact. And we were almost at a loss to know what to do. It was obvious that noradrenaline was not working at the almost industrial doses that we were using, neat noradrenaline, and we would normally use 1,000th of the dose. And so I was aware of the study, the availability of LMM, and she seemed just the ideal candidate to try it on. So I contacted Patrick and said, you know, I think this would be a suitable person and a valid reason to do it.
They did give us a percentage in the region of 80-20 against. So our chances of survival was 20% roughly. And we was in a figure, I suppose, within hours of her life.
If the theory was correct, the infection had kick-started some dormant NO production system in all the wrong cells inside Jean Gregg, and her blood vessels had relaxed almost beyond control.
We went down there, treated her with the protocol that we designed because we wanted each patient to get exactly the same doses in exactly the same way so that we could perform some sort of statistical analysis and see whether it was really working. I think it's very important that these things are done in a way that allows analysis and not done in some random ad hoc fashion.
We introduced the drug very cautiously at a low dose, 0.3 milligrams per kilogram, and amazingly enough, perhaps we were expecting their response, but tremendously for Gene, we were able to get her blood pressure up, and it actually did have a response, which is tremendous. So we did it again, because we wanted to make sure it was just a real phenomena and not something that happened, and the same response happened. We got an increase in her blood pressure, and so we went to a higher dose, and then an infusion, and we were able, on the infusion, to bring down the noradrenaline dose, which to a much more respectable concentration, such that she was able to perfuse her fingers to a certain extent, certainly much better than she was before. But the most important thing was we maintained her blood pressure.
I can remember looking down and seeing all these black toes and thinking, how on earth did that happen? And then I looked at my fingers and they were all black, and I could see black here going both-side. It just seemed remarkable, and all these needles here. I thought, crumbs, what have they done to me? I thought, you know, I've had a new drug, but not the first in the world to receive a new drug. It's just marvellous to know that they can use it on other people now, and that it saved my life.
I've always wanted to do research that in some way would be applied to a medically relevant, to a problem of real significance. And you always like to think that that's going to happen. It is no small motivation for what I do and the kind of problems that I choose to work on in my research. But I never thought that it would happen so fast, especially when we were still in the midst of working on it, because applications of basic findings often occur years after the basic findings. The fact that the applications are occurring now, I think, is just one indication of how important the field of nitric oxide research is.
Jean Gregg is living proof that our bodies need to dice with this dangerous molecule to survive. But hers is just one of many medical conditions that promise to become clearer since the physiologists and biochemists struck gold. Rather than acting as a poison, NO has been controlling parts of our bodies since our ancestors crawled out of the mud. There is hardly a field of medicine that isn't now investigating its role. In the lungs,
it's becoming apparent that it has a very important role if inhaled. Inhaled nitric oxide is another, it vasodilates the lung tissue as well. So it acts as a vasodilator in that situation. And it's very important in septic shock associated lung disease, ARDS as it's called, and also in neonates and premature babies too who have respiratory distress when born.
In cases of brain damage.
Well, apart from stroke, which is the one I've mentioned, there are a number of other possibilities where too much nitric oxide may cause pathological changes in cells. And so people are looking in diseases like Parkinson's disease, in Alzheimer's disease, in multiple sclerosis, and a whole host of others, just to have a look. Well, what's happened to what's happening to the nitric oxide system in these conditions? Is it possible that there's too much of it being produced and this is a factor contributing to the disease?
NO is also essential in allowing blood to enter the penis to achieve an erection. The gas finally hit the headlines.
For several weeks, the phone in my office and in my laboratory was just bombarded from patients who were phoning in trying to determine where they could buy this nitric oxide so they could relieve their problems of impotency. The incidence of impotence in men in the world is very high. It's estimated to be roughly 9 or 10% of all males have some form of impotence. And so you can imagine how many individuals were concerned about that.
Mm-hmm. Mm-hmm. Mm.
When something new appears, we don't believe that it exists. Once it's demonstrated that it exists, we wonder why we missed it for so long. And I think that's a natural process. When you think how many functions nitric oxide is playing, it's really amazing that we hadn't seen it before.
I think that one of the things that's most interesting about the story is that the early work was in a field that no one, I think, would have seen as being of direct relevance to clinical medicine. And a good example of that is the work done on how the bull retracts its penis. I mean, who would have thought that that would be relevant to human heart disease? I certainly wouldn't have.
So Professor Gillespie's mystery chemical was NO. The NANK nerves that release the bull's penis and do so much in our own bodies use NO to do it.
I was amazed. I had never, ever conceived that the substance I was interested in would be nitric oxide. And even more, I never believed that nitric oxide would have a part to play in the body. So I really believed that it would be either in the peptide, fatty acid, or maybe a carbohydrate. But the idea that it should be an inorganic compound and one which we knew to be quite poisonous, it really would never have occurred to me.
Professor Gillespie has now retired, but not before he saw Dr. Martin return after several years in blood research to work again on the mysteries of the retractor penis muscle.
Laboratory work is always quite intriguing because you feel sure that the breakthrough, the key observation, is going to be made quite soon, though the years pass. But nonetheless, you always feel that quite soon you'll get the answer to that.